So, good evening. Hello, everyone. I would like first to introduce myself. I'm Liu Dong. I'm the director of Microbiome Innovation Center of GemPharmatech. And this night I'm very happy to have this webinar and discuss the topic of germ-free mouse models and their applications in microbiome-related research. And I really hope that tonight you can enjoy this talk. Let's go. Screen's freeze. Just a sec. Okay, here we go. So, this microbiome research has experienced major technical, practical, and conceptual process in recent years. Especially during the early days, the initial focus was to describe the composition and diversity of microbiome and to identify correlation between microbes and phenotypes. However, current research now is already shifted from the correlation towards refined study dedicated to gaining like the real functional insights into the microbiome. Now describe its mechanisms of action and studying its co-evaluation with the host. So this ongoing effort can advance our understanding of this microbiome influence of the various aspects of host, which we can find big on the right side and showing the sports in microbiome research. So the association of human health with the intestine has been long acknowledged. The human body is colonized by a diverse of bacteria, viruses, archaea, and also other occurrence. Microbes like inhabited all human body surface, but a significant, but a, sig- I mean, but a significant number of microbiome live in the gastro tract. Let's also say in our gut called gut microbiota. It is widely accepted that approximately like let's say a trillion, 100 trillion microbiome live on and inside the human body and having a key role in various biological processes, including the health and also the disease. That's the primary mediator of human homeostasis and impacting various biological activities such as metabolism, also the barrier function and inflammation through both intestinal and also the extraintestinal action. So this is gut microbiota also recently been let's see, called or classified as a vital organ because of its multi-directional connection to the axis with the organ through the neural, endocrine, humoral, immunological, and also the metabolic pathways. So any changes in the microbiome community not only cause gut-related issues, but also can influence other organ-related disease. So murine models actually, are very indispensable tools for researchers to study the relation between gut microbiome and human health and disease. So since like let's see, if I remember correctly, so it's since 1960, the specific pathogen-free model. Let's also see the SPF mice model has become the golden standard for the laboratory animal research. So the history of this model and the advantage of using animal free of pathogen for advancing biomedical research has been reviewed extensively. However, the SPF model has some major pitfalls that shouldn't be overlooked. For example, these pitfalls are also part of the reason for the diversified methodologies used to study host and microbiome interaction in vivo. Fortunately, we currently have more choices than ever before when it comes to designing and also selecting an appropriate level of microbiome diversity for colonization of our laboratory rodents. So gem-free rodents are such a useful animal model for investigating the far-reaching influence the microbiota on host responses. That's in the following slides. I would like to first introduce the application of germ-free mice and their role in microbiome research. Follow up to that, I would like to also introduce fecal microbiota transplantation in germ-free mice, because fecal microbiota transplantation is one of the most frequently used methods to study the interaction between microbiome and disease and health. And thirdly, I would like to introduce, maybe you might be less familiar with this model. It's called gnotobiotic mice model, and also talk more about their application in microbiome research. And lastly, I would like to introduce our germ-free and microbiome service platform, and to discuss what we can do and how we can accelerate your microbiome researches. So in the following presentations, the talk will be divided into 4 parts. So first, I would like to introduce the definition of germ-free mice. So what is germ-free mice? Actually, germ-free mice are laboratory animals that lack all life organisms. These organisms including like all microorganisms and parasites and viruses and everything that determined within the limitation of the detection methods now currently available, which means the current detection methods cannot detect anything on germ-free mice or in germ-free mice. So this is the definition of germ-free mice. And actually the concept, this is the concept of the germ-free mice animal was put forward more than a century ago by Louis Pasteur. I think, yeah, he's very famous in 1885. At that time, he believes that survival without any bacteria is impossible. However, 10 years after that, so it's 10 years later, it's 1895, I think scientists from Berlin University produced the first germ-free animal. It's a guinea pig, but it's only survived for 13 days until, and after like half century, it's till 1946. So the first germ-free rat was established in 1950. And isolators were invented, which is light, practical, and also, let's say, this is a house for germ-free mice. So after the house was built, the first germ-free mice was successfully developed in 1959. So this is history and how we found the germ-free mice. And actually, because germ-free mice and SPF mice are currently two most being used in microbiome researches. But what is the differences between germ-free mice and SPF mice? So actually, because germ-free mice lack of symbiotic microorganism from after they birth, so there indeed have some differences between germ-free mice and SPF mice from anatomical characteristics and also the physiological metabolism characteristics and also the development of their immune system. And I would like to elaborate them one by one. So first, in the term of anatomical characteristics, the body weight, the heart, the lung, another lung, and the liver, the kidney, spleen, and syncon is different between germ-free mice and SPF mice. What did we do? And actually, we compared the different organ of germ-free mice at different ages with that of SPF mice. As we can see, there are so many organs are different between those two, but one of the most notable characteristics of germ-free mice is their cecum. The cecum of germ-free mice is most likely have like 3 or 4 times bigger than that of SPF mice, and this is due to the lack of gut microbiota in germ-free mice. So germ-free mice is unable to digest fiber. So the fiber will, let's see, the food debris and also the non-digestible fiber will stock in their cecum, so making the signal on germ-free mice is much bigger than that of SPF mice. So this is characteristics of the autonomy term of germ-free mice. And in addition to that, actually, because germ-free mice are also absent of enteric microorganism flora, and this can definitely impact the development of the immune system of germ-free mice. Let's see. So here we actually measured 3 different antibodies of germ-free mice and SPF mice at different ages. So we found that no matter on 8-week and 80-week germ-free mice, the IgA level is lower than that of SPF mice. And we also found that the IgM level of germ-free mice at 80-week germ-free mice is a little bit higher than that of SPF mice. So actually, the development of their immune system is actually very different due to the differences of their microorganism. So this is a bit about their antibody and also their immune system. And adding to that, we also measure the hepatic and also the renal function indicators. What we found here is the germ-free mice at 80 weeks have significantly lower blood AST level than SPF mice, and also the GF mice at 8 weeks have significantly higher blood AKT level than the SPF mice. So which this also showing that the metabolism characteristics of GF mice is different with SPF mice. And next, I would like to emphasize that because germ-free mice and SPF mice is very different, and when we use germ-free mice, we need to bring that into our mind and to have this sense that germ-free mice is very different with SPF mice. However, because germ-free mice is also one of the has been extensively reported or used in investigating the interaction between microbiome and diseases. So it can and during those kind of studies, when you want to study the relation between microbiome and health and or let's say diseases, fecal microbiota transplantation is the most important, is one of the most important tool for doing that. So I think everyone knows that that germ-free mice can be used in different diseases, and this is why I'm going to introduce the most important or being used tool, which is fecal microbiota transplantation. The application of fecal microbiota transplantation in germ-free mice in the part 2. So actually, what is fecal microbiota transplantation? So first, we would like to isolate the fecal microbiota from the feces. It can be both from the mouse, from the human. It really depends on what kind of study you are conducting. So first, we would extract the fecal microbiota from feces, and then we will do the fecal microbiota transplantation to germ-free mice via intragastric administration. And after that, we would do the efficacy evaluation. And after efficacy evaluation, we need to collect their feces because it's really, but it also depends on what kind of study you are conducting, and we can you would pick the time point to collect their feces you are interested in. So after you collect their samples, you need to do the in vitro function analysis. Either can be sequencing and also maybe coupled with other measurements to do the in vitro function. So actually, this is a comprehensive procedure of how we use the fecal microbiota transplantation in our GPT system. So what we actually did and what I want to show you here is 2 cases of fecal microbiota transplantation, because actually we did collect the feces from mice and isolate the fecal microbiota and then do the FMT to germ-free mice. This is first, and then we also isolate the human feces microbiota and do the transplantation. We indeed see the differences between these two scenarios. This is why I want to show this to you. And here's the first cases is that we isolate the mouse microbiota from mouse feces. Here we first isolate and we do the FMT 3 times as a first week. And after that, we'll collect the feces and do the 16 SRN epicon sequencing at different time point. And at the last time point, we also do the tissue collection and did the HE staining to see whether there are some changes of the phenotypes. So what I want to emphasize is here, after different times, the gut microbiota of those recipients will change. So what does this mean? So here, as I just showed here, we first isolate the feces microbiota and do the transplantation as a first day. We consider that as a day 0, and we do the same FMT at day 2 and day 6. And here we see after the first injection of after the first FMT, we can see actually the observed OTU and also the channel index can recover the alpha diversity can recover after 14 days of the recipients when compared with the feces microbiota. So on the left panel, you can see the observed OTU recovered after 14 days, and also the channel index recovered after 14 days, and also the Simpson did a similar thing. But when we see the beta diversity of different time points, actually the day 14 is the beta diversity of day 14 is similar to that of day 21, but still, even these 2 time points is very close, but it's still far from the feces microbiota. So what does this tell us is that the alpha diversity of the recipients. So the recipients means we did the FMT to the different mice. So the alpha diversity of the recipients is can recover to the level of feces microbiota after 14 days, but the beta diversity is hard to recover. So this is what I want to show you, and that was fecal microbiota transplantation from mouse, and here I also want to show you cases of the human microbiota transplantation from human, because in the most cases we do isolate the feces from human and transplant the feces microbiota to germ-free mice to see whether its phenotype can be recovered in a germ-free mice model. This is what we frequently did. So here we also isolate the feces microbiota from human feces and transplant those feces into the germ-free mice and see what the differences, or let's see what's microbiome stability changes over time. So what we did here is first we isolate fecal microbiota from human feces, and then we transplant those to germ-free mice. And after 2 consecutive injections, we collect their feces at different time points, which is week 1, 2, 3. So we collect their feces every week, and also do the 16 SRN epicon sequencing over time. And what we found is here. So actually, we did the transplantation based on 2 different mouse backgrounds. So the first is black 6 germ-free mice, and the second is Barbie C germ-free mice. Why I do this, because I'm afraid of the background would influence inoculation of the gut microbiota in their gut. So I supposed to do the comparison. So what we found is here is do have some similarities. So what we found here is after the transplantation of the microbiota, after 1 week, the observed OTU has dropped 1/3. So you can see at the top left, the observed OTU of the feces species is around 300, and after 1 week FMT, the observed OTU dropped to around a little bit above 200. So it's dropped to 2/3. But after the first drop, you can see the composition, or this is observed OTU is keep a very stable status. So this is why it happened both in black six and also the PBC. I guess is because when you do the transplantation of the feces from human to germ-free mice, because if different species, of course, some of the microbiome from humans is hardly inoculated in the gut of germ-free mice. This is due to the species different. So after those species that cannot inoculate in mice from human in mice, it can drop directly. And after that, what left in the mouse guard, it can keep growth. So this is why the alpha diversity is very stable over after the first week. So when you see the week 1, week 2, 3, they keep a very stable status. Both it happened both in black six and also BBC. So this is microbiome composition stability after the fecal microbiota transplantation from human to germ-free mice. In addition to the alpha diversity, what we found here is also very similar to that in the FMT in mouse. So the week 5, week 4, week 3, week 2, week 1, so the beta diversity is changed over time. And what is also similar to the last cases is that both of this time point is very far from the fecal microbiota. So it's the same. Even so after the first week of fecal microbiota transplantation, the alpha diversity keep a very stable status, but the beta diversity is far from the fecal microbiota suspension. So this is also told us when if you want to design your experiments, if you want to use fecal microbiota transplantation from human to germ-free mice, you need to choose a proper time point. This is what I want to show here. And here I also summarize the general loss in black six over time and also in BBC over time. This is to tell you like whether your humanized strain or humanized species can or cannot inoculate in the gut of germ-free mice. And actually, because as I already mentioned twice, I think fecal microbiota transplantation in germ mice is one of the most frequently used if you want to investigate the relation between microbiome and researches. It has been applied into so many diseases, for example, like the aging, the IBD, and also obesity. So what we already did for our clients, this is one paper has been already published, which is called a study on the methods and effect of a construction humanized mice model. What our clients did is they give us feces from patient. It's not it's not a patient actually from a pregnant, and then we use mice and extract the microbiota from the feces, and we construct the humanized gut microbiota mouse in germ-free mice, and we compare the models if we use germ-free mice and SPO mice to see which is a better model to construct the humanized gut microbiota mouse model. And the conclusion term says the better conclusion is that like we use germ-free mice that can construct a better humanized gut microbiota mouse model when compared with SPF mice. And 21 days of FMD is the best time point. this is the study our clients published. And after that, I would like to also introduce a little about our research work. It's called we actually established the model. It's called gnotobiotic mouse. And I want to talk about the use or this is the application of this gnotobiotic mouse in microbiome researches. And actually, what is gnotobiotic mouse? Actually, gnotobiotic mice I think, is less familiar for you guys because it actually has a long history, but it's not famous as germ-free mice. So actually, gnotobiotic mice are laboratory animals that carry known or specific microbiome antibodies, which are mainly obtained by colonization individual bacteria or flora into germ-free mice. So actually, we can also consider the gnotobiotic mice is also derivative germ-free mice. It just like colonization of specific microbiome. But why we use gnotobiotic mice? Because I also mentioned before, like SPF mice and germ-free mice are two of the most frequently used mice model in microbiome research field. But why we use gnotobiotic mice? Here, let's see. So because germ-free mice actually are not stimulated by microbiologisms from birth, so the phenotype and also some of the functional germ-free mice are different from those of SPF mice, right? So, however, if we colonize some specific microbiome to germ-free mice, mixing gnotobiotic mice, they can recover some of the phenotypes and functions to the level of SPF mice when you have also meanwhile when the phenotype and the function recover to the SPF mice at the same time you also have a very clear microbiome background, so they can reflect a more realistic physiological status, and also it can become a better model, or let's say a relatively better model in the study of microbiome flora mechanism. So this is why we want to use gnotobiotic mice. But how we construct our gnotobiotic mice by ourselves actually is a little bit complicated. First, we use large numbers of strains, and we transplant all the strains into germ-free mice, and then we make them natural screening, and we make them populated and see what is left in the germ-free mice. And after the few times of natural screening, we also covered with some bioinformatic analysis, and at the end, we have established the mouse with six different microbiome strains, and we call this kind of gnotomices GMM6. So it's with the six different microbiome strains, and this is a list of the six different microbiome strains of this mouse, and you can also see the abundance of these six different strains. And on the right, you can also see the distribution of the abundance of these six different strains in feces, colon, cecum, and small intestine. So this is abundance of those different strains, and we also see that the abundance of these six strains did not have big differences between female and male mice, which means the gender would not be influencing factor. And here, what we characterized after we colonize these six strains, we also measure the phenotypes. Here we see the analogical, here we see that all GMM6 organs, except the cecum, almost recovered SPF mice model. Actually, I want to elaborate a bit more here. What we did is first we have SPF mice, then we colonized the GF mice with the six strain. It's called the GMM6, and then we also have GF mice. So we compared those three different mice models, and we see no matter it's the organ things and also the body weight, recover quite great, right? When you see when you compare the SPF mice with GM6, it's rarely recover very great, of course, except the second. After that, we also measure the fat weight because everybody knows that the microbiome play an important role in the fat metabolism, and we also see that there are differences between the fat percentage of SPF mice and gene-free mice. And however, we can see on the left top, after the colonization of 6 strains and also in our GMM6 mice model, the fat ratio is recovered quite good, even though they do have a significant differences with SPL mice, but when you compare GMM6 with gene-free mice, the fat ratio increased quite a lot. And also when we talk about the metabolic things, we found there are no differences in the lipid metabolism index of GMM6, and also the TH was only slightly lower than SPF mice, which means the lipid metabolism index for GMM6 recovered quite good. And also because microbiome or this microbiome indeed influence the development of immune system. So we compared the immune index of these 3 different mice. Here, what we found, we actually measure 6 parameters. We measure the leukocytes, which is MCD45 positive, and we also measure CD3 positive T cells, CD19 positive B cells, and CD5 NK cells, monocytes, and neutrophils. Actually found except except NK cells, the proportion of the immune cells in their blood actually of GMM6 actually all recovered to a SPF level. And we also measured the index in the spleen and also the antibodies. So here the conclusion is that also immune index and also organs and also other things recover quite good when they compare to, recover to a very good SPF level. So it's quite a good model for study the mechanism of those things. Here, actually, there are also clients already use our GMM6 models to do the publication. Actually, we are also the co-author with of this paper because actually we also contributed some work of the things in addition to the GMM6 models. This paper has been published on Nature Metabolism 2 years ago on 2022. So also use our model to see whether the high protein diet can prevent the fat mass increase after dieting by contacting lactobacillus and lipid absorption. So they do use GMM6 in that paper. So if you are interested in this paper, you can just search this paper after the webinar. I will not dive into too much details of that paper. So last, I would like to also introduce a little bit about our GemPharmatech and microbiome service platform. And I introduce this is to really want to show you what we can do to accelerate your microbiome researches. So here, actually, because I mentioned quite a lot the interaction between, or this is the relation between diseases and microbiome. But how did the researchers in microbiome field do the study? What is the roadmap? So here, as you can see here, first, the roadmap for microbiome researchers, first, they would like to collect the feces from different sources. It can be both like mice, can be also from human. First, they collect their feces, and then they use a multi-omics analysis to compare the differences from their like healthy donor and patient and or healthy mouse or these models they first will do the omic analysis to see the differences. After that, they would like to culture or let's say isolate some microbiomes they found that they found differences in the previous step. So they need to get the real microbiomes rather than only get the data. So after they get some microbiome, they can do the functional screening. And then they will see whether this different microbiome really plays the expected function. So they would like to put the microbiome into the disease mouse model and see whether the microbiome can really elevate the diseases. And after that, they would use different mouse model to do some mechanism studies. And then they would also show, because here you will find out the answer of whether the microbiome is related or associated with the diseases. And after that, it's not the end. You also need to identify whether the microbiome is a cause rather than association relationship to the diseases. So they need to do the cause effect by using like germ-free mice or biotic mice to see what is the cause relationship between microbiome diseases, and then they will do the customized sequencing, and after which they need to do the kill function and also the pathway analysis. And then they can have wrap ups all the results and do the publication. So actually, this is a roadmap for the microbiome researchers. And what we do and what we can help with the researchers is that we use our animal models and also some in vitro resources to help them with the whole roadmaps. So what we actually doing is we offer a one-stop solution for the microbiology researchers, which can be classified at different states. Here, first in vitro, we can because in the in vitro part, the microbiome isolation is quite difficult. So we have offered our mice and human we built up mice and human microbiome library. So this can be used to counter the difficulties in microbiome acquirement in the in vitro part. And in the in vivo part, we actually offer the mice models with different microbiology background, which we can offer both SPF mice and antibiotic treated mice, germ-free mice, and autobiotic mice. We can do that. And we also have a 6 ex vivo analysis platform to do the also ex vivo platform. So wrap up of this three part, we actually do have the one-stop solution for the microbiology researchers. So here I would like to elaborate a little bit more for the microbiome part. Also for the in vitro part, actually we built up our microbiology plan for like 3 years ago. So what service now we can do is first we can do some microbiome strain culture, isolation, and preservation. And then we can also do the fecal microbiota transplantation in our microbiology platform. Then we also have the mouse and also the human microbiome library in our microbiology platform. Here is mouse microbiome library, which you can also. This is only partially listed. You can also find this list in our official site. Here we already isolated 145 species and also covered 80 in general. And in addition to the mouse microbiome library, we also have the human microbiome library, and you can also find in our, I think it found by requirement. So now we already have more than 120 species. So if you here why we build up those microbiome library is to, if you have some interested microbiome you found in the list, you can just tell us. We can directly pick those species and do the following animal experiment. So it can save you tons of times. This is for the in vitro part. And for the germ-free mice part, we actually have we actually have a lot of germ-free mice. We have two kinds of germ-free mice. So first is what we have is off-the-shelf germ-free mice. The live products we have is the general used black 6 germ-free mice, the Barbie C germ-free mice, and also ICR. And we also have the from preserved products like the in immunodeficient germ-free mice model is also called NCG germ-free mice model, and also the IL-10 knockout model, and also the NCG knockout model. So if you want to like some gene engineered mice model with a gene-free mice version, we can also customize that by in vitro fermentation. In vitro fertilization. So we can get any gene engineered mice of gene-free version if you want. So this is partial. This is a gene-free mice product we have. And during the in vivo part, this is combination of the mice model and the microbiome. So here in this part, we have the isolators for the race and also for the experiment execution for the whole microbiome surface experiments. So what we can do here is we do some we did quite a lot of the following things. For example, we did a lot with single strain or flora colonization, and we also did quite a lot of fecal microbiota transplantation, and we can also build up a lot of different disease models in our in vivo platform. We also do the efficacy evaluation and also strain safety assessment in our in vivo platform. But at the end of the in vivo experiment is definitely not the end of your experiments because you also need to do the in vitro analysis to get some data. So we also coupled different, our platform also coupled with different ex vivo analysis platform. So the first way, because we are also the IMPC compliance, we can do a lot of phenotyping analysis. For example, we can do the phenotyping of metabolism, behavior, and also the blood panel and extraction, etc. Then we also have our own flow cytometer analysis platform. We can use dose analysis, CBA cytokine assay, and etc. And we also can do some molecular and cellular things, for example, DNA, RNA extraction, and also gene staining. So actually, we have different platforms, different ex vivo platforms in order to make sure different experimental requirements. So actually during different status of your researches, you indeed need different mice models. So we actually can offer different mouse model, not only from the different microbiology background, but also from different diseases. What you can find if you are investigating the efficacy or this is a functional studies of your microbiome, we can offer different mice from different diseases. So it really depends on what kind of studies you're doing. You will pick up the right mice model. And then when you're doing some mechanistic studies, we can also offer the mouse model with different background, the different microbiology background. So if you are doing the mechanistic studies, you might need the gene-free mice, and if you are doing some to check the association of the microbiota with diseases, you might also need some antibiotic treated mice. So we actually want to offer all the mice that suit for all the stages of your researches. So here are the publications of our clients using our services and also using our gene-free mice. So now we already published like 14 articles, and all those articles are quite high level, and we have Cell, we have Science, we have Nature. So all of those papers has high qualified. Now we have curative impact factor around like now it's already around 400. So to conclude, actually what we did is one-stop solution for the microbiome researchers. So we have our production plan for actually give you the choices of different mice models, and we have our microbiology platforms to give you the resources of microbiome and also the opportunity, the platform to do the transplantation or to the isolation, those kind of stuff. And then we have our in vivo platform to do the real experiments. Then we also coupled the ex vivo platform to do the analysis to get the data. So this is how we can help you with the whole process of your microbiome researches. So this is talk today. Then I hope it can really help you a lot.